talking about nutrients. This is the fourth lecture of the Chemical Oceanography module. So there are more than one major nutrients in the ocean. The two that we tend to think of as being the most important or the sort of major nutrients are nitrogen and phosphorus. But there are lots of other things known as trace elements that are required, but they're required in smaller quantities than nitrogen and phosphorus. Other important ones include silicate for certain things such as diatoms, and then a lot of trace metals, just like we need trace metals, the biology in the ocean needs trace metals. So when you talk about major nutrients, you're really talking about quite a few different things that are used in different ways by different species and are required in different concentrations. So nutrients is a very general term, but we're going to talk quite a bit about nitrogen. A nutrient by definition is a chemical needed to support photosynthesis and primary production. That's the sort of oceanographic definition of a nutrient. And here on the bottom right, you can see there's this nice schematic of ocean nitrogen cycling. And it's worth understanding how nitrogen moves around the ocean, the different forms that are present, and how it interacts with different phytoplankton and bacterial species. So from the dissolved gases lecture, you know that N2 gas is the most abundant gas in the atmosphere, and it equilibrates with the surface ocean. So there's dissolved N2 in the surface ocean. And you might think that's great because organisms need nitrogen. The atmosphere is absolutely chock full of nitrogen. But the reality is that this dissolved N2 gas is not really usable by most organisms in the surface ocean. There's a handful that can use it through a process called nitrogen fixation, where they can convert that dissolved N2 into ammonium. But that's a really specialized process, and it really only happens where it's warm, where the surface waters are very stable, and where organisms can use light to break that really strong triple bond. Most nitrogen forms that you find in the ocean are actually as this species, nitrate. So nitrate is used by phytoplankton. That's why you see this arrow going this way here. So phytoplankton use nitrate. They also use ammonium, which is a different nitrogen source here. And then those phytoplankton that are using this nitrogen as a nutrient, ammonium and nitrate, they get consumed and move up through the food chain. But what ends up happening is these phytoplankton, as they die, they end up being converted back to ammonium. And the same with other things higher up in the food chain. And when you convert them back to ammonium, they go through a process whereby they can be nitrified, so converted in the ocean from ammonium to nitrite to nitrate. And so all of these different nitrogen forms are sort of cycling through. There's different little bacteria, archaea, phytoplankton, higher organisms, all using these different forms of nitrogen in different ways, sometimes even recreating these forms of nitrogen. And this is known as the nitrogen cycle in the ocean. Ultimately, nitrogen doesn't keep accumulating in the ocean. It does eventually go away, or we'd end up with an ocean that was just full of nitrogen. And there's a loss process or a removal process is denitrification. And there are bacteria that conduct denitrification in certain regions of the ocean. And what they do is they take nitrite and nitrate, and they actually reduce it back to N2 gas, which ends up leaving the ocean. So that's why it doesn't build up, and you just don't end up with tons and tons of nitrogen nutrients in the ocean. So because nitrogen isn't completely building up in the ocean, and other nutrients also aren't building up in the ocean, there's this idea that something must be limiting the growth. And so this is where we get to the concept of Liebig's concept of the law of the minimum. So the phytoplankton in the ocean, if they have enough nitrogen, they'll just keep growing. But eventually they are going to run out of something. So it might not be the nitrogen they run out of, it might be something else that they need. And so the extent of growth of all organisms in the ocean will eventually be limited by the supply of something, whether that's a major nutrient like nitrogen and phosphorus or a micronutrient like iron. And when you run out, growth stops, and that's known as the law of the minimum. And so it's really important to understand which regions of the ocean are limited by which nutrients. And one way to look at this, so if you look at the top left plot, this is a view of the surface ocean, And these are, the colors indicate the concentration of nitrate. So that's the most abundant form of the nitrogen in the surface ocean, or in the ocean in general. And that's one of the major nutrients. And the scale here is micromolar, so that's 10 to the minus 6 moles. So you can see in vast areas of the ocean, it's purple. And purple is essentially not a lot. You can see way down here, it's very low. Meaning there's not a lot of nitrate in the surface ocean. There are some major exceptions, though, as you can see, where the values, for example, in the Southern Ocean down here, values skyrocket, so 35 micromolar. And in these regions here, up in the subarctic North Pacific, also very high values, again in the Arctic. And then there's this really interesting region here in the eastern tropical Pacific where you also see somewhat higher nitrogen. And then if you look down at the bottom right plot, this is an image of surface chlorophyll concentration. So surface water chlorophyll concentration, this is a way of looking at how many plants there are in the ocean. So how many things are growing in the surface ocean. And you can see, interestingly, that there's a lot of stuff growing in the Southern Ocean and a lot of stuff growing in the northern high latitudes as well, in the North Pacific. And in some ways, there's sort of, if you look at these two maps, it appears as if there's some sort of a relationship between the two of them. Okay, so what's controlling? Why is there not a lot of nitrate in the surface ocean? So essentially, why is most of the surface ocean purple here? So we'll move from looking at nitrate in the surface to looking at it with a depth profile. So now here on the right, we've got with depth looking down here, and then our nitrate concentrations across here. Okay, so in this case, values at the surface are getting much lower as you go. I don't know why this is the number 20. That should be 0. I don't know what's going on there. But values get really, really low at the surface, and that's because they're being consumed. So in the top surface layer here in the euphotic zone, you've got light. And so when you've got light and you've got nitrate and other nutrients around, phytoplankton are going to use them, and they're going to keep using them essentially until they go away, which is why you see really low values in the surface ocean. But then when you go below the euphotic zone to the deeper parts of the water column, everything that was alive in the surface sinks, and it becomes remineralized, so converted back into its dissolved forms. And so that's why the concentrations increase with depth. And one way that we say this is to say that nutrients are non-conservative. So non-conservative meaning that the concentrations of the nutrients vary regionally, they vary seasonally, and they vary here with depth, as you can see. The vertical distributions in different basins look different. And this is something that'll make a lot more sense once you start to learn about thermohaline circulation. And so what ends up happening is if you look at the right at the nitrate plot, in both the Atlantic and the Pacific, values get really low at the surface. And so in both ocean basins, those nutrients are being consumed. And what you see as you go down to depth, where the remineralization is happening, that it looks like the values are much higher in the Pacific than the Atlantic, and they are. And there's a clue as to why this is if you look at the left on the oxygen plot. So the process of remineralization is essentially respiration. Respiration consumes oxygen. So when you have a lot more respiration happening, then you would expect there to be less oxygen in the waters because it's being used. So you can see here there's less oxygen in the Pacific, and that's because there's more respiration that's been happening than the waters in the Atlantic. So there's been this water as it cycles through, it goes through the Atlantic Ocean first, and then everything gets respired, and you produce this nitrate and you consume some oxygen. And then that water keeps moving. It keeps going into the deep Pacific. And so you produce more nitrate by consuming more oxygen through this respiration process. And so you can see that in these depth profiles. And so what we say is that the waters that end up in the Pacific are older than the waters that are in the Atlantic because they move through the Atlantic basins first and then get to the Pacific. And you guys will talk about this a lot more when you get to thermohaline circulation. But it's interesting to note that this process, one of the ways we know that remineralization is driving this sort of deep signal below the surface, is because you get this mirror image between your oxygen profiles and your nitrate profiles. So here again, this is another example of essentially the same thing, where these are very classic dissolved nutrient profiles showing that there's an increase in the deep water concentrations from the Atlantic to the Pacific and the Indian. And here you can see again, you've got depth on the y-axis, and this is showing on the left phosphorus. This is the first time we're looking at phosphorus here. But essentially the profiles look identical to the nitrogen profiles, not in the numbers. You can see the concentration of phosphorus is much lower, but the shape that they have over the course of the water column is essentially the same. And so you would say that phosphorus also has a nutrient-like profile. So it goes completely to zero in the surface, as does nitrates. And then the remineralization, so everything grows and consumes the nitrate. It uses it all up because there's sunshine and everything grows. And then it sinks, it dies, and bacteria start to remineralize it and respire it, and the dissolved forms come back out. And you end up with seeing the nutrients again. Here again, looking at depth profiles, and now moving on from nitrate and phosphorus to looking at there's phosphate here and silicate. So phosphate's in blue, silicate's in green, and you can see silicate as well has a nutrient-like profile. And then again, your oxygen is basically a mirror image. It's exactly opposite, and pH is doing the same here. And so that's because, as you can see in the text here, so I'm using some of these words interchangeably, but essentially decomposition, bacterial respiration, remineralization, they're all the same thing, okay? So it's anything that's growing in the surface, so phytoplankton, any essentially organic matter, here it's referred to as particulate organic matter, is really what's controlling the depth profile of what these nutrients look like. Here, these are from the subtropical Pacific, and they show how the nutrients in the oxygen are sort of backwards mirror images of each other. And this is quite typical for most ocean basins and for most of your major nutrients. This schematic here shows the slight differences between nitrogen cycling in the open ocean on the left and the coastal ocean on the right. So the coastal ocean is a little bit more complicated. So we'll start over here on the coast, and you can see in brown here, this is your coastline. So at the coast, you have to worry about things like riverine inputs of nutrients, so nitrogen, phosphorus, and iron. And you also have this process of upwelling, which you guys will talk about later in the course when you get into more the physics of how the water is moving around. But upwelling brings with it deep water. And deep water, as we just learned by looking at those profiles, has a lot of nutrients in it. And so this upwelling process at the coast brings nutrients in. And this schematic on the right is very much what it looks like if you know anything about the Benguela. It's an upwelling system on the west coast of South Africa. And so you bring lots of nutrients in from these rivers and from upwelling, and you end up with these nutrients cycling through really rapidly. So you've got lots of ammonium, lots of nitrate, and you end up with so much growth. You can see all the green here. This green is supposed to symbolize all of your plants, your phytoplankton growing. And they eventually die. And so that's your organic matter here coming down. And there's so much of them that they consume all of the oxygen in the water. And you end up with what's called an oxygen minimum zone, where there's so many nutrients and so much growth, and then they all die and end up using so much oxygen that you deplete the water of oxygen. That's quite different from if you look out in the open ocean on this side, where you have your much smaller, less phytoplankton, so you see less green here. It's also a little bit lower, this in the water, so it's a bit deeper. And what that is it's where this, so on the left-hand side, these are your depth profiles. So this is showing you depth here, where you have light in the surface ocean is nice and high, and where you still have some light and you start to have some nutrients, that's where you have your growth in the open ocean. And so you need both of these things. You need the nutrients and the light to be present. And so you have your organic matter, and again here it's consuming nitrate, nitrite, and ammonium, and it sinks and is remineralized, but there isn't quite as much of that as you have over here in the oxygen minimum zone, where you've got all of this growth. So there are differences in the cycling of nutrients between the open ocean and the coastal ocean. And when you get into biogeochemistry next in C2005, you'll get into this in a lot of detail. And so one other thing I wanted to discuss during the nutrient lecture is this really fascinating concept known as the Redfield ratio. And so what happened is this guy named Redfield a long time ago started compiling all these measurements of nutrients in the ocean. And what he found was that pretty much anywhere, wherever you are, you always found nitrogen and phosphorus in the same ratio. And this is now known as the Redfield ratio. So the elemental composition of different organic molecules, they vary quite a bit, right? If you look at the amount of carbon and nitrogen and phosphorus in carbohydrates, it's very different than in proteins. But if you sum up across all of the different kinds of molecules in organisms, for living material in the ocean, the molar ratio of carbon to nitrogen to phosphorus is pretty consistent regardless of where you are. And that's what this number right here is, the Redfield ratio, 106 to 16 to 1. Whoops. 106 to 16 to 1. And so what you can see here is that, so during photosynthesis, organisms use CO2, and they use water, and they use nitrate and phosphates, and they're consumed in this ratio of 16 to 1, where you can see 16 HNO3 plus 1 phosphorus molecule. And so that's what makes a model organism. And on the right-hand side, when you have respiration, you end up having this sort of model organism is degraded back to its dissolved components, and you end up creating this here, your 16 to 1 nitrogen to phosphorus. So when you go out and measure phosphorus here on the x-axis and nitrogen here on the y-axis in the water, you measure, let's say you measure around 16 for nitrogen, it's around then 1 for phosphorus, right? But if you have less nitrogen, you have less phosphorus, and it stays in the same ratio. So this line is your 16 to 1 line. And all of the data, regardless of where you are in the ocean, are sort of falling around this. And this is sort of remarkable. And it's something that's used a lot in biogeochemistry. Because what it means is if there's ever a situation where the data don't fall along this line, you can learn a lot about the processes that are happening in terms of what's growing, what's not growing, what processes are taking place or not taking place. So this is a pretty brief introduction to nutrients, but it's important because we're going to soon be talking about primary productivity. And before we talk about primary productivity, I just wanted to introduce you to the basics of nitrogen.